Welcome to the IGIS webinar, Research Update and Discussion on the Important Role of MSG for Sodium Reduction. My name is Ana San Gabriel. I am the Science Advisor and the member of the Secretariat for IGIS organization, and I will be the moderator for this webinar today. As you are, I can see that many participants are joining, so I will go a little bit slowly at the beginning. I have some announcements to make. First, if you have questions, please use the Q&A box to write your questions. For your information, we are going to be recording the webinar, and I would please ask you not to screenshot the presentations. We will provide you with the shareable documents when you need them. This is the agenda for today. Today's agenda, I will introduce first the speakers. You will see their presentations, and then next we will have a 10-minute session where you can ask questions through the Q&A box. Let me just introduce you a little bit for those of you who are not familiar with the International Glutamate Information Service. We are a non-profit organization, international non-profit organization, and we have 7 members throughout Australia, Asia, Europe, and North and South America. Our goal is to provide up-to-date evidence based in information about glutamate, monosodium glutamate, MSG, and Umami. And I'd like to take this opportunity to thank the speakers for accepting to participate and all the IGIS members who have supported us for having this webinar. So I would welcome the speakers maybe to activate their cameras so that you can see them. Hi Gary, hi Sue, how are you? So today's webinar will focus on why and how to use MSG as a sodium reduction tool and its impact on public health. We will have these 2 experts that are accompanying me right now in the screen. One in the field of chemical senses, Dr. Gary Beecham, who will talk about the taste of salt, why we like it so much, and what role MSG could play when reducing salt. And another expert in the field of food and sensorial science, Dr. Suyu Lee, who will show us her latest research on the role of MSG for the acceptance of low-salt foods. Gary Beecham is a distinguished member and the Emeritus Director and President of the Monell Chemical Senses Center. As you will see in his presentation, he has dedicated his career on the study of the biology of the smell and taste perception, especially sweet and salty taste, including umami, of course, with the aim to improve nutritional health. Dr. Suyu Lee is professor at the Department of Food Science and Nutrition in the University of Illinois, Urbana-Champaign. Her research focuses on the utilizing innovative sensory methodology to develop health-targeted new products, alternatives, and promote lifelong healthful eating habits. So with any further ado, let's listen to their talks.
Hello, my name is Gary Beecham. I am a member of the Monell Chemical Senses Center, and I'm going to talk a little bit today about salt taste. First, though, I want to say something a little bit about the Monell Center. The Monell Center is a basic research institute that is dedicated to research on the chemical senses, by which we mean smell, taste, and some other touch senses, such as that stimulated by hot peppers or menthol. Our goal is to improve the health and well-being of humans by advancing scientific understanding of these senses. And as a basic research institute, we conduct highly disciplined, multidisciplinary basic research. We disseminate new knowledge through both basic scientific publications and for publications for the popular media. We create and license intellectual property. We assist in translating research for commercial purposes, work with many companies. And most importantly, perhaps, we train students and scientists from government, industry, and academia in the chemical senses. This is a photograph of our two buildings. We're located on the edge of the University of Pennsylvania campus, and we're loosely affiliated with the University of Pennsylvania. We have more than 50 PhD scientists with expertise in a wide range of scientific disciplines, biochemistry, molecular biology, genetics, and so forth. And basically, we are an interdisciplinary group that studies these senses that are both chemical, biological, biophysical, and really span all of biology. This is the part of the body that we're most interested in today, where we first receive the information from odors up here on the top of the nose, up around the odor perception area. Tastes are mainly, at least from the perceptual point of view, in the tongue and in the palate, although there are taste receptors actually throughout the body. And then irritants, such as hot peppers, can be detected in the mouth, in the nose, and even on the skin throughout the body. So two of my most favorite white powders are sugar and salt. These are sort of chemosensory superstimuli in the sense that they drive intake and have been extremely powerful in stimulating excess intakes of both calories and of sodium. What I'm going to talk about today is salt, sodium chloride, and particularly focus on the issue of excess salt intake. But for both of these, both for sugar and for salt, it turns out that they are really involved quite centrally in a variety of very important diseases that are seen around the world. This includes obesity, diabetes, hypertension, in particular for salt, hypertension and high blood pressure, resulting in heart disease and many, many deaths, and even some cancers. So these stimuli are very, very important for looking at human health. So a brief outline, first I'm going to talk a little bit about salt and health, very little bit about salt taste perception. Then I'm going to talk about strategies to reduce sodium in the diet, the various strategies that we can use to hopefully reduce the amount of foods we consume, and the amount of sodium that we consume in particular. And then I want to go on to the University Institute of Medicine findings. So let's begin at the beginning for humans. For many humans, sodium has been historically very difficult to find. It's obviously can be found in seawater, it can be found in mining areas, but for many parts of the world, it's very rare, and it's absolutely necessary for life. If you don't have sodium, you die. So for many eons, people use sodium as a way of exchanging value. Sodium, the word salary, comes from, in the right here, where I'm showing this magnificent golden piece of the crown jewels of England. This is the salt cellar, and this sort of demonstrates the importance and power of salt throughout the world. Now, it used to be, as I say, very difficult to get sufficient salt in many places in the world, and humans fought wars over it. But now we sort of have the opposite. We have too much salt. Salt is now extremely cheap. It is almost, aside from water, the least expensive thing you can put into foods. As a consequence, in part, we consume way, way too much sodium relative to what we've been evolved to consume during most of our evolutionary history. So it's been said that once a person reaches his 50s, he or her is in the risk of developing high blood pressure over the remainder of the lifespan. Up to 90% of those people will develop this high blood pressure. And it's been estimated that by reducing it in the United States, we prevent 100,000 lives and annually save billions of dollars. So this is a really important and big public health issue. How do we go about reducing salt in the diet? Many, many government organizations from throughout the world, not just here in the U.S., but in many, many countries, and this is an example of the World Health Organization, have recommended across the board reductions in sodium in the diet. WHO recommends reductions to less than 2 grams of sodium a day. That's 2,000 milligrams. And frankly, most people, certainly in the developed countries, consume way, way more than that. Sometimes 30, 40% more, sometimes twice that much. And so this is the issue. How do we reduce it to a level where it's less dangerous to human health? First, let's start about salt perception. How does it work? Basically, if we think of the tongue, which I pointed out earlier, tongue has these little bumps in the tongue that are taste buds. And this is an example here of a taste bud. And the taste buds are made up of taste cells, and each cell is specialized to detect a particular taste quality. These are the so-called basic tastes: sweet, sour, salty, bitter, and umami. So this cell detects sour, this one detects sweet, and this one detects salt, which is what we're interested in today. On the top of each cell are receptors, and perhaps along the sides as well. And these receptors are the ones that can detect, in this case, sodium, or in the case of sweet, sugars. Here's a putative salt taste receptor. This is basically the sodium would come here to the receptor area and would be transduced a signal into the cell. And then this salty cell, this blue one here, would have a neuron that goes back to the brain and signals saltiness. Remarkably, this, I say, a putative salt taste receptor because we're not so sure exactly how salt taste works. It's amazing that given all that we know about the biology of food and taste and flavor, still don't quite know exactly how salt taste works. But we do know that there are specific cells that send this message to the brain. Now, salt is really a magic ingredient and is actually gives much more than saltiness. And this is a very old study, which is a beautiful example of this, looking at the taste of a plain chicken soup. And the way this slide can be read is this is the center, and it's the distance between the center and, say, this point says how salty the plain soup is. So this is unsalted soup, the gray. So the unsalted soup is this salty, but if you add salt to it, which is the blue, it makes it much saltier. Of course, that's true. But what's interesting here is it not only makes it more salty, as you can see, but it also increases the fullness. It increases the sweetness of the food, in this particular case, the soup. And very importantly, it actually reduces these off tastes, metallic and bitterness. So if you add salt, you get a reduction in sort of bad tasting things. So salt, when added to a food, makes it not just salty, but it makes it much, much better. We've been studying for years the interaction between bitter and salt, and what we've been able to show is that these bitter compounds, so this is quinine, caffeine, these are all bitter compounds of various substances, these two being acetaminophen and pseudoephedrine, which are drugs. They are bitter. Almost all drugs are bitter. Along this axis, we've measured how bitter these tastes are when actually suppress them with salt. So the reduction in bitterness that salt can induce can be very, very high. Let's just look at urea, which is this one right here. It reduces it by 80%. So many bitter compounds are made much less bitter by adding salt to them. Now, what this does actually, in a very simple-minded way, is it can make things much better, as I showed you in the slide before last. So if we take sucrose, which is pure sweet, and again, we're measuring here, this is the sweetness of sucrose. It's pure sweet, which is in blue, but it's very, very low in bitterness. And then we consider another substance, which is urea, which we showed before was very, very bitter, but also not very sweet. But if we add these two together, so we mix them, but keep them at the same concentrations, what happens is they suppress each other. So we have both a sweet, bitter mix, but neither one of them is as sweet or as bitter as the original stimulus. Now, if we add salt to that, and here I'm talking about not just salt, but sodium. So the sodium ion, we can do this in a variety of ways without making something salty. So we're not adding something salty to it, we're just adding sodium. And what that does is it inhibits the bitterness, knocks down the bitterness, as I just said, and as a result, allows the sweetness to increase. And so in this way, one can show that you can take foods, add salt to them, And by reducing the bitterness, you actually make them sweeter. And so this is one reason we believe that sodium is so commonly used in so many foods. We don't know really how this works. And again, this is an area which is surprising, that we don't know how the sodium actually inhibits bitterness. And the thought is that if we could develop ways to inhibit the bitterness without adding sodium, with some other substance, that we could use this as a technique to reduce bitterness and reduce the sodium in foods and make those foods still taste not bitter and sweeter. And so we at Monell have a large project trying to identify other bitter inhibitors that we could use instead of sodium. Now, there's other potential strategies to reduce salted foods, and I want to talk about three classes of them actually. One is the idea that you could develop salt enhancers. This would be substances that would increase the saltiness of food with salt in it, but not by adding sodium. A second is salt substitutes, things that we could substitute for salt that provide saltiness, but would not have as much sodium. And the third is a really different approach, which is to make people consume less salt and adapt to that. And I'll get to that a little bit later. First, let's look at a salt enhancer. This is arginine, which my colleagues have demonstrated is a salt enhancer. If we look over here, this is the saltiness of pure salt with nothing added to it, with arginine added to it. With arginine added to it, and at two different concentrations. And as you can see, even when you add arginine to pure salt water, which is what this is, no salt, it doesn't give much of a saltiness to it. But if you take sodium chloride at a higher concentration, which is salty, and add arginine to it, you not only make it salty, but you make it much saltier by adding the arginine. And here this shows it even greater. So arginine itself is not salty, but when you add arginine to salt, you can enhance the saltiness. And so many people are trying to find these various salt enhancers that are both safe and that are effective in causing lower levels of salt to taste saltier. Another approach is to use odors. It turns out that odors that have been associated with foods that are salty take on a perception of saltiness. So it's possible to increase the saltiness of a food if you can add an odor to it, or a flavor that has been associated with saltiness. Now, for salt substitutes, the go-to substance that has been used throughout the past 50 years or so is potassium chloride. Potassium chloride tastes salty, which is, again, another puzzle we have because we don't really understand how it is that potassium chloride stimulates salty taste, since we think sodium is the ion that really is doing that in sodium chloride. But in any case, it works. And it turns out there are many salt substitutes using potassium chloride that are mixed with 50-50 sodium chloride to reduce the amount of sodium that's consumed. However, this is not a perfect solution because many individuals find potassium chloride, even when mixed with salt, to be very unpleasant tasting. They describe it as bitter or yucky or just bad. And so this is not a perfect solution, but it is one that's being used by industry to reduce salt in foods. We also know that added MSG can result in lower sodium intake under certain circumstances. This is going to be discussed in much detail in the next presentation, but I just wanted to briefly go over the background for this. If you'll remember this slide, this showed how salt itself adds not just saltiness, but fullness, flavor strength, balance, reduces off tastes. It turns out that MSG does much the same, has much the same effects on food and food preference. I take this very, very old set of data that was really just a very early meeting amongst people in the Army and that were interested in food in 1948, who had a discussion as to what the flavor of MSG really was. I have some quotes from that that are, I think, relevant today as they were in 1948. You can read them down there, but I want to go to the bottom two and read them specifically. The persistent effect of MSG is present in the whole mouth. It is difficult to describe this sensation other than to call it the feeling of satisfaction. And what I've got in red here, the closest similar taste I know is salted chicken broth. So in essence, what these people were saying is that, like salt, MSG has a whole range of flavor characteristics, not just the umami taste, but also the flavor of the substance, the feel of the substance, the mouth feel, and so forth. So this results in the fact that one can reduce the sodium, again, in chicken soup, for example, as shown on the left here, by about 30 to 40% or 50%. Add a small amount of sodium glutamate. So it's adding a little bit more sodium, but much less than you took out. And these two will be equally palatable. And as I said, this will be shown in much more detail in the next presentation. But basically, the finding is that MSG can replace not only the saltiness, but also the other flavor characteristics of salt within a food. And for this reason, it is a very highly efficient way to reduce salt in the diet. Finally, I want to get to the other approach, which is to not change the food, but to change the person. And this comes from anecdotes and clinical ideas going back at least 100 years or maybe more that suggested that when people are consuming a diet that's relatively high in salt, and for whatever reasons they decide to go or put on a low sodium diet by their physician, they first find the food just terrible. It's just too bland. It doesn't have that salty, wonderful taste to it. It just is very unpleasant. But it turns out, according to these anecdotal reports, that if people stay on that lower sodium diet for a period of time, which is probably 2 or 3 months, their frame of reference shifts downwards. So things that used to taste just right now taste way too salty, and things that were not salty enough now taste just right. In other words, we can shift their frame of reference down so that what they prefer in food is actually less salt. The first study that really demonstrated this was one that was done by my colleagues at Monell, which demonstrated that when people were tested with crackers, and these crackers have various levels of salt from this low level to a relatively high level of salt in crackers, and this measures how much people like these crackers. Before they went on a low sodium diet, basically the rule was the saltier the better. So the highest level of salt was the preferred one. After they'd been on the low sodium diet for 3 months, what we found was that this level of salt that used to be preferred now is liked much less, and the optimal level of salt is actually half of what they wanted before. This observation has been repeated many times. This just shows some papers that have studied this, and it is well believed that this is a phenomenon that is true for most people and probably for everybody around the world if we could get people to do salt reductions. So based on this, 2005 and eventually published in 2010, the Institute of Medicine of the United States set out a conference to discuss strategies to reduce sodium intake in the U.S. And I've sort of gone over some of the ones that they've suggested, but the main one that they have suggested is the one that I've just mentioned. They saw that the taste and flavor of foods that salt provided was the biggest problem. And they also noted, and this is work originally also done at the Monell Center, that in the U.S. and in many developed countries at least, the vast majority of salt that we consume is consumed in foods that are not made in our house, that we don't cook, that we don't add salt shakers to, but are manufactured by companies and in restaurants. And this just illustrates the percentage of salt in various foods. And what we're seeing here is that salt is everywhere. It's in all the foods that we purchase, both in restaurants and in grocery stores. And so as a consequence, the idea of reducing salt in the diet really can only work if we can reduce it in foods across the board. And so as a result of this observation, what the Institute of Medicine recommended to the U.S. government was that the Federal Drug Administration, FDA, set mandatory standards to require food and restaurant industries to gradually reduce salt content in their products. This is a very strong recommendation. The mandatory part of it was of great concern, and in fact has not been instituted. But the idea has been, and this idea would be that if we could get industry and restaurants to gradually pull down the level of salt in foods, because it would be gradual and because people would adapt to it, it would not make the foods less palatable, and people would eventually, in the population as a whole, come to prefer lower salt in foods. So this is the sort of the diagram that was put out in the Institute of Medicine report that basically suggests that over a period of time, and this is period of time down here, they have this stepwise reduction that industry and restaurants would gradually lower their salt in foods. And if this was done in small increments over a period of time, maybe 10, 15 years, one could shift the whole population down such that there would be less consumption of sodium and the health of the population as a whole would benefit. So what's happened with this? Well, in 2010, the Institute of Medicine recommended these mandatory gradual reductions. The government never accepted that, but they did mandate reductions along the same lines as in a stepwise way for sodium in foods for children. Unfortunately, this policy, although it was mandated, has not been followed well and is really stalled. In 2016, the FDA proposed voluntary reductions of salted foods, and so there is a large database proposing how industry should gradually reduce the salt in many, many foods. And many companies have seriously taken this as a goal and are working hard to do it. But quite frankly, the push from the FDA and from the federal government was reduced. And even today, we don't have the finalized goals the next several years. So the hope is that given different political climates, seeing the importance of this, that this will happen and there will be a much stronger push to reduce salt in food. So if we can think of what's the hopeful vision for the future, we start with the idea that much of the U.S. and world population is consuming sodium at levels that are simply too high to be safe. It's not really a safe food environment. There are multiple strategies that we can use to reduce sodium intake using salt enhancers, salt substitutes, as well as this strategy to gradually reduce the salt food in manufactured foods. If we could do this, the overall reduction in sodium intake will benefit human health, and we believe it's true in the United States, and we believe it's probably true in many, many countries around the world. This is my last slide. I just have presented a disclosure. The source of funding for my research is the research of my colleagues, which includes support from the Ajinomoto company. And finally, I thank you for your interest and I look forward to hearing some questions.
Hello. Thank you for inviting me to present in the International Glutamate Information Service webinar. Today I would like to share my presentation that covers the use of flavor enhancers to aid in sodium reduction. For the remainder of the presentation, I would have my camera off to enhance the usage of the slide space. Overconsumption of sodium is a global issue. As it can be seen on this map, in 2010, the sodium intake among adult males was far above the World Health Organization recommendation of 2,000 milligram per day, which is in the blue zone, and you don't see many of the blue countries on this map. And in fact, the mean global intake of sodium was close to twice the WHO recommendation and was about 4,000 milligram per day. In 2010, Institute of Medicine published a report on the strategies to reduce sodium intake in the United States. And in the publication, they describe 3 broad categories of research needs on sodium reduction. And they are to understand how salty taste preferences develop throughout the lifespan, to develop innovative methods to reduce sodium in foods while maintaining palatability, physical properties, and safety, and to enhance current understanding of factors that impact consumer awareness and behavior relative to sodium reduction. As a food scientist, my main focus of research had always been to develop innovative food technology methods to reduce sodium in foods. And for this presentation, we will focus on using flavor enhancers such as monosodium glutamate in sodium reduction. When we look at the source of dietary sodium in the U.S., approximately 30% of it comes from restaurant, fast food, and home foods. The remainder, 70%, comes from processed foods, among which the top categories of source of sodium would be breads, grains, cereals, and then red meats, poultry, eggs, mainly the cold cuts and cured meats, and dairy products such as cheeses. My research group has conducted a scoping review on sodium reduction strategy and found 277 primary studies that were relevant to the topic. Within it, we discuss the categories with the most research conducted regarding sodium reduction, categories of food, in fact. And what we found was that protein foods, primarily cured meats, was the highest category that was investigated, and then followed by grains, the bread, rolls, and tortillas as the second highest food category that has been researched. And then third, the milk and dairy products, namely cheeses. And this aligns with the food categories that is the top sources of sodium in the U.S. In our scoping review, we also reported on the top strategies that are used for sodium reduction. Number one we found was the salt replacers, and then followed by salt removal, where the salt is removed without adding any other replacers or doing any modification to the food product. And then the third is the flavor modification. And this is where the flavor enhancer category would reside in, followed by physical modification and functional modification. So within the flavor modification category, which comprises of approximately 30% of the studies, you will see the flavor enhancers as the strategy for sodium reduction. And as you can see in this category, the top foods that are investigated under this category is the protein foods category, followed by the mixed dishes, and then we have the grains, the breads, and bakery goods, and then followed by the cheeses. When we look at the flavor modification category much more specifically, we see that number one category within it is umami ingredients, followed by glutamate strategy, such as using monosodium glutamate, and then the third amino acids category. And what we note is that these three most common are all associated with conferring an umami taste. So let's dive into a few of my research that has been published in the area of using flavor enhancers for sodium reduction. First one, which was published in the Journal of Food Science in 2019, is on consumer acceptance of reduced sodium potato chips and puff rice, and how we are able to reduce the sodium to the level that FDA recommended, yet still maintain consumer acceptance through the use of flavor enhancers. And the second publication in the same area was in the Journal of Food Science again in 2020, and it's in conjunction with consumer acceptance tests. We had done trained panel descriptive analysis test to identify the drivers of liking for these reduced sodium snack products using flavor enhancers. Recognizing that approximately 90% of Americans consume more sodium than the recommended limit of 2,300 milligram per day, which can lead to various chronic diseases such as hypertension and heart diseases, in 2016, FDA has drafted a voluntary sodium reduction recommendations for packaged, processed, and prepared foods. And they had two level of reduction goals, the short-term 2-year goal and the long-term 10-year goal. In our study, we had two different snack products. One was the puff rice product, and then the other was the potato chip product. And for the puff rice product, the 2-year FDA goal was about 20% reduction, and 10-year FDA goal was 57% reduction. For potato chips, the 2-year short-term FDA goal was 18% reduction versus the 10-year FDA goal was 51% reduction. In our publication, we clearly present the background information about how the flavor enhancers such as monosodium glutamate and disodium inosonate and guanylate are safe, and they contribute to savory taste and flavor that can remedy the issues with sodium reduction. And we also provide information that they're found in natural foods such as Parmesan cheese, tomatoes, and mushrooms. The aim of our study was to compare acceptability of reduced sodium potato chips and rice puffs seasoned with and without flavor enhancers across 3 different conditions. In a blind tasting condition where the panelists are not able to see the ingredient information, and then the second condition where ingredient information is given to the panelist, and then in third condition, ingredient information is given, followed with the education piece. As seen on this slide, the experimental design consisted of 2-day sessions. On the first day, the panelists came in to do the blind and informed conditions with 5-minute break in between. On the second day, they came back to do the informed with education session, followed by demographic questionnaire. This is the educational piece in its entirety that the panelists had to read during the informed with education session. It consists of the need for sodium reduction and the FDA guidelines for sodium reduction, both for the 2-year and the 10-year goals, and how the liking decreases in reduced sodium products. And then it ends with how flavor enhancers such as MSG and I+G are safe to use and that they provide a great solution to increase liking of reduced sodium products. In our experimental design, for both potato chips and rice puffs, we had 7 samples, a full sodium sample, and then reduced sodium short-term and long-term without flavor enhancers, reduced sodium samples with flavor enhancer MSG, and then reduced sodium sample with MSG and I+G. For the short-term FDA goal, the amount of sodium that is contained in specifically the potato chip sample that is shown here was around 0.18 grams, and then the long-term was around 0.11 gram, which was about 50% reduction from the full sodium. And for the full sodium sample, it would comprise of 9.2% of daily value of sodium, and then the short term would comprise of 7.5% daily value, and then the short sodium with long term sodium reduction would contain about 4.6% daily value. Potato chips were processed by purchasing no salt added original potato chips, and they were heated in an oven for 30 minutes at 93 degrees Celsius. And then they were sprinkled with barbecue seasoning and seasoning with salt, MSG, and I+G according to the experimental design, and carefully shaken in a Ziploc bag. And then, voila, we had the potato chip samples. For puffed rice, we purchased brown rice, and we extruded the rice in an extruder to create the rice puffs. And then we season the rice puffs with spice seasoning and various salt and flavor enhancer combination according to the experimental design. And then use a tumbler to coat the seasoning and the salt. And voila, we had the puff rice. The consumer tests involved a vertical 9-point hedonic scale, which is depicted on this slide here, to fit the mobile phone and tablet format for evaluation. And we used Qualtrics as the software to acquire the data. The attributes that we used to have consumers rate was overall liking, saltiness liking, and flavor liking. And the location was a study all type of setting. For the descriptive analysis session, we used a hybrid form of spectrum and quantitative descriptive analysis. There were 8 sessions that were used to familiarize the samples and derive attributes and references, and 4 sessions that comprised of attribute definitions, reference selection, and sampling and rating procedure. And then there was 1 session where the panelists practiced the session in sensory booths. And then we had 2 sessions to have the actual rating done and to provide duplicate rating of the same samples. This is the potato chip attributes, definitions, and references list. And as you can see, under the modality of aroma, we have the meaty and raw potato attributes. And under the aroma by mouth modality, we have the cooked potato and meaty attributes. Under the taste modality, we have the salty and sour attributes. And then under the aftertaste modality, we have the fried potato, meaty, salty, and umami attributes. This slide shows the reference foods that were used for the potato chip attributes. So, for example, under the aroma modality, we had the meaty and raw potato attributes. And for the meaty attribute, we used real bacon bits. And then for raw potato attribute, we used raw potatoes. This is the list of attributes, definitions, and references with their reference intensity for the puffed rice samples. As you can see, under the aroma modality, we have the paprika attribute. And this is because we used spicy powder that contained paprika for the puff rice. Under the aroma by mouth modality, we have the chili powder attribute and the roasted attribute. And this is also because of the seasoning that we used. Under the texture modality, we have the crunchiness attribute, and under the taste modality, we have the salty and savory attributes. And finally, under the aftertaste modality, we have the bitter, savory, and garlic attributes. This slide illustrates the reference foods that were used for puff rice evaluation. And as can be seen, under the aroma modality, we had the paprika attribute, and we in fact used paprika as the reference food for that attribute. For savory attribute under the aftertaste modality and taste modality, we used actual MSG solutions. So our findings were that flavor-enhanced samples with MSG and I plus G included were rated the highest in the blind rating condition in the consumer test. And in fact, the ingredient information increased the liking of reduced sodium and savory samples, which was contrary to our hypothesis. We hypothesized originally that the ingredient information may decrease the liking due to the information of reduced sodium as well as including MSG in the product. But in fact, it increased the liking. Further, education increased the liking or maintained the liking from informed condition levels. And when we added the descriptive analysis findings to the consumer test, what we found was that savory, salty, garlic, and crunchy are the drivers of liking for these snack products. The last study that I would like to highlight in my presentation is a study done by a group of researchers at the University of California at Davis. The study was published in the Journal of Food Science in 2020. In the study, the researchers used monosodium glutamate to mitigate the loss of sensory palatability of salt-reduced products, and mainly those food products that were deemed better for you. So the aim of this study was to compare consumer acceptance of reduced salt versions of 4 so-called better for you dishes enhanced with MSG through a salt flip in an amount that still substantially reduced total sodium to the normal salt versions. The experimental design consisted of 4 recipes. Cauliflower fried rice with pork, denoted as CR, roasted vegetables, denoted as RV, quinoa bowl, denoted as QB, and savory yogurt dip, denoted as SD. The participants of the study evaluated these 4 different recipes in 3 different versions. Standard recipe with typical salt content, a reduced salt recipe with significant sodium reduction, and third, reduced salt recipe with significant sodium reduction plus MSG, resulting in slightly higher sodium content than the reduced salt recipe with significant sodium reduction without the MSG. So table 1 shows the sodium level for each recipe and percent sodium reduction for the standard recipe. So as you can see, the reduced salt recipe ranged in salt reduction from around 47% to 63%. And then the MSG recipe was slightly higher than the reduced salt recipe, but still the range of reduction in sodium was from around 30% to 60%. And as you can see on the right-hand side, how all of the dishes looked in terms of the visual appeal. The consumer participants rated overall liking, appearance, flavor, texture, saltiness, aftertaste, and how likely they would be to order the dish at a restaurant for each version of the dishes. And the findings were that MSG recipes were associated with delicious, flavorful, balanced, and savory. And some MSG recipes were liked as much as or better than, in the case of quinoa bowl and savory yogurt dip, than the standard recipes. Reduced salt recipes were commonly described as bland, and the standard recipes described as salty and sour in some cases. So overall, MSG containing recipes were more appreciated by the consumer panel. The overall conclusions of my presentation and some thoughts for future direction for research in sodium reduction and using flavor enhancers as strategy are as follows. So we found that flavor enhancers were effective in mitigating the loss of sensory palatability in reduced sodium snack products and healthful products. We also found that education was effective in increasing liking for foods with flavor enhancers. There is room for future direction in identifying other products that would benefit from sodium reduction and that we could use flavor enhancers as the strategy in which to mitigate the loss of sensory palatability. Another area of future direction is to identify the mode of education that may be more effective and further enhance consumer learning to accept flavor enhancers such as MSG. With that, I would like to thank the organizing committee for inviting me to present at this webinar, and I would also like to thank you all for your kind attention.
So I would like to go further with the Q&A. Just a second. Here I am. Okay. Hi. So Gary, you can show your face if you want. So it seems that most of the so far there are many questions coming, but I think that the first question that came was about safety. But not only about safety, but there is an interesting one also that ask why there is different standards of reduction of salt. So the WHO talks about 2 grams, the FDA talks about 2.3 grams. I don't know, Gary, do you want to comment about that?
Well, there is no doubt that there are differences in the recommendations, but they're all in the same direction, and they're all pretty much on target, I think. The idea that there's excess salt consumption overall is universally accepted. Well, I wouldn't say universally, but the vast majority of the people who have worked in this area believe that that we're consuming throughout most of the world excess salt. The exact numbers, of course, are really difficult to pin down. But if we could reduce the amount of sodium consumed by 30 or 40%, there is no doubt that there would be many lives saved, a lot less heart disease, stroke, and what have you. And so I think that there's the issue of the exact amount is really a false issue. It's just, it's too much, and we have to find ways to reduce it.
And can you give one sentence about MSG safety? You don't, I mean, just...
I have not studied MSG safety, but I've worked with MSG for 40 or so years. And from what I can tell, the research demonstrates universally that MSG, when consumed in normal amounts, is entirely safe. There, like everything, there may be a few people who are sensitive. We can't rule that out. And those that are obviously shouldn't consume it. But it is the data that I'm aware of suggests that as a population level, MSG is as safe as certainly safer probably than sodium chloride.
Thank you. I will share some resources later on for if somebody wants to check in more detail. So there is also I realized that you have a different concentrations of MSG depending on the recipe. So somebody's asking, how much MSG we have to use and what would be safe. But can you comment about the amount of MSG that we use, even in your experiments?
Sure, yeah. I can't give a definite amount because actually it depends on the food and the serving size. So I think if you look at some of the recipes that calls for MSG, and just looking at the U.S. cuisine, you can see like meat dishes calls for a teaspoon or half a teaspoon of MSG, which is close to that 2,000 milligram of sodium. That's how much they're calling for. But it's a serving size of, let's say, 4 to 6 people in a meat dish. But if you're using MSG, for example, in my recent research on bread products, it's calling for much less, and it's going to be much more effective and potent in aiding with sodium reduction. So you can reduce up to 50% of the sodium of regular use, but then use very small amount, maybe less than even 100 milligram. And still it would be effective because you're not calling for that enhanced umami flavor. And I just want to dovetail on what Gary said about the safety of MSG is that it is recognized as generally safe. It's a GRAS substance recognized by FDA. So really science has shown that it is safe to use. And there are people who are sensitive to MSG, but also in the research field, you see in the literature that when you have a placebo and a real research design of seeing the sensitivity. It's not called for consistently either. So people might think that they're sensitive to MSG, but they may not be. So there's that kind of a contradiction too, but it is really generally recognized as safe.
Thank you for mentioning the study. So actually, the double-blind placebo study. Actually, there is one question where they also for you. So they can believe or they can see with your experiments that you can go away with 25 to 40% reduction of salt. Do you think you can go to 50 to 60%? I think some of the recipes went as far as 50, the one that was using pork, but what do you think? Or Gary, if you want to say something about that too.
Well, just to answer that question, that also depends on the food product. And I feel like I'm answering questions in a vague way. But if the salt is called for other functionality in food, for example, safety. So salt has been used a lot as a preservative, right? And in a bread system, salt also enhances the gluten activity. So it aids when the rising effect of the bread. So if it's called for other functionalities, there is a limit to how much you can reduce. So I believe if the salt is only used for flavor and taste, you can go down to 40, 50, 60%, even lower, and then substitute it with MSG. But if salt is used for other functionalities, it may not be possible to go so much in terms of lowering, the reduction percentage. So it depends on the food.
And just to say a good word about salt, we can't live without salt. We have to have salt. And so there have been concerns about people going too low. I think that's very, very rare. And in our own experiments, when we put people on very low salt diets, the body's remarkably good at retaining sodium and adapting to a low sodium diet. But nevertheless, there are instances where people lose sodium, and it's a very serious issue. So you don't want to go to zero. For both sensory reasons and for physiological reasons, having and consuming salt at lowered levels is a healthy thing to do.
I know neither of you are doctors, but they asked me, would it be okay to have MSG for a high blood pressure person?
So
it's a little bit difficult to answer.
I think they're asking if hypertensive patients can have MSG. And I just want to say MSG is glutamate, which is glutamic acid. It's an amino acid, and sodium. So MSG actually contains sodium, like the salt contains sodium chloride, right? So there's a percentage of sodium that you would consume with MSG. And the studies have shown the glutamate in MSG versus glutamate in natural sources is really not distinguishable, and the body processes the same way. So my understanding, although I'm not a doctor and a nutritionist, I believe that, in an amount that is acceptable for hypertensive people to consume sodium, MSG can be safe. And then just recognizing that MSG contains sodium. It's not free of sodium.
Yes, I understand. I'm going a little bit out of time, but I would like to continue if the participants don't mind. There is a question, Gary, about if MSG is such a good option to reduce sodium in the industry, in the food industry, why it's not being so widely used? What do you think?
Well, I'm not so sure that it's not being widely used. It's just not being widely labeled, I think. There are ways to get sodium glutamate, umami flavor and umami contents which doesn't say MSG on it. So if I go back, I think it was remarkable, actually, the last set of studies that Sue talked about the reduction in sodium and the enhancement by MSG, and think about the list of quartermaster comments that I put up from 1948, and they were identical, really. How the MSG added not just saltiness, but added the mouthfeel and the feeling of fullness and what have you. We don't understand how this works. And to me, as a biologist, that's the really interesting question, is how is it that these stimuli actually cause these things that are not just the perceptual saltiness, but actually the feeling that your mouth is full, and it's a flavorful thing. You reduce the blandness in ways that are still not understood. So, from a research point of view, to me, that's still a very interesting topic. But it is one that I think is remarkably consistent across studies, from beginning in the 40s and 50s and 60s, and now into real good controlled studies that are looking at how we can reduce salt in specific foods. And I think that also was an important point that Sue was making, that it really depends on the food.
Yes, yeah, actually, I think that it's one of the most important conclusions of her presentation today. So what about when you gave the education to your participants? Do they have the education about the properties of MSG as an enhancer?
Yes.
Did they understood that? Okay.
Yeah, so the slide that I showed for the educational piece was in its entirety. That's what I gave to the panelists as a whole thing. And so there were 3 parts. First, how there's a lot of sodium consumption, over sodium consumption, because really the recommended amount by the FDA is 2300 milligram for regular population. But it's an interesting 90% of adults are pre-hypertensive and hypertensive in the US and consuming a lot of sodium. And when you're in that range, you're supposed to actually consume 1500 milligrams. So there's a lower upper range for at-risk population. And it also depends on ethnicity. So there's a lot of components to these amounts. But anyway, the average amount that is consumed is 3,400 milligrams. So it's really much higher than what is recommended, and double the amount than what's recommended for at-risk population. So with that, that's an important message to deliver. So that was the first one. And the second one was, when you reduce sodium, people don't like it. That was kind of the second message. And so to mitigate, there are flavor enhancers, and the educational piece introduces that, and monosodium glutamate and I+G that we used in our study is those flavor enhancers. And then we give the background information of how, in literature, the scientific literature, it shows that it is safe to use, and it's recognized as, generally recognized as safe substance by the FDA. So that's kind of what we gave as the education. But the reason why we had the 3-step study was we were hypothesizing, expecting that the liking rating will go low when the ingredient was revealed, but in fact it didn't. It increased. So it was interesting, that was a surprising result. And then with the education, it maintained the liking, or it enhanced it a little bit. So education component wasn't
when they understand. Yeah. So it seems important. So Gary, I have 2 questions about salt for you. Do you think that we like it so much, so it's kind of a addiction? It creates addiction?
I have a slide that says the original addiction is to salt. My view, and this is teleology, it's really sort of evolutionary speculation, but throughout history for humans, and actually for many species that eat plants where sodium is low, there's been a real necessity to find salt and get it. And in fact, there's a strong drive if you're sodium deficient to detect and consume salt in large amounts, and this has been studied for many, many years. But presumably, during evolution, there's no drive to stop consuming salt. Why would you worry about it? Because as we were both saying, the development of hypertension and blood pressure issues comes at an age when people are no longer reproducing, and actually most humans died long before that time. So it has always struck me that to build an animal like ourselves to seek out and consume salt and to like it, it makes sense. And there's no reason to put a stop to that. And so in our society, where we've got excess salt and where salt is so inexpensive, of course we will consume more than probably is needed. And so that's where the problem lies, is that we're built to go after it, that it causes pleasure and has these remarkable characteristics from a sensory point of view, and there's no particular reason from a biological point of view for us not to consume as much as we want. There is now, because we would rather live to be 70 or 80 than to be 20 or 30, which was what most humans lived not so long ago.
That's right. Well, we have so many questions, and I have questions myself, but the time it's running up. So let me, close today's webinar. I will encourage, if we can answer some of the questions by mail, we will do that. We appreciate everybody's participation. Let me just share with you that besides the Glutamate International Glutamate Information Service, which is the website in the left, you have other resources to check about Umami, MSG, Glutamate, from MSG Facts or No MSG. Even the Umami Information Center has a lot of resources about Umami. Those of you who are worried about safety and need to have a more reassuring information, you can go to the Mayo Clinic. They have a page specialized in MSG, and also the US FDA has a Q&A about MSG, so that you can see what will be the standard acceptance about the safety of MSG. But I'm staying in Japan, so I wanted to give you a sense of the summer here right now in June with a lot of hydragens. I want to thank everybody for your attention, for participating, for your questions. And if you have anything to ask specifically or contact us, please go to the glutamate.info @gmail.com, and we will be there to help you as much as we can. Thank you very much, everybody, for today. I think it's a topic that we can be talking for hours. Unfortunately, time is limited, so that's all we wanted to share in the webinar today. Thank you very much. Thank you, Gary.
Thank you.
Well, thank you very much. It was a pleasure.
Yeah, it was a pleasure to collaborate with you too. I learned a lot. Thank you.